Hello, everyone. Welcome to Brain to Brain. This afternoon, we're continuing our week-long series of programs with brain science researchers at the University of Minnesota. I'm Amber, the Public Science Events Manager at the Bell Museum, and we're excited to collaborate with these scientists and learn more about their work in the fields of psychology, neuroscience, and neurology. If this is your first time joining us this week, you might be wondering why we're focusing on brains. Well, in addition to the incredible research that's being done at the university, the Bell Museum is excited to announce the debut of our original production, Mysteries of Your Brain. This new animated film will be showcased all weekend long in our planetarium, and it will become part of our regular viewing schedule. We hope you'll join us in person soon to see this immersive show. Before I introduce our special guests, I'd like to share that we will save some time for questions this afternoon. If you have any, please post them in the comments and we'll get to as many as we can. I'd also like to take a moment to thank Boston Scientific for their generous support of brain-related programs and activities at the museum. And now I'd like to introduce this afternoon's guest. Dr. Uzma Samadani is an associate professor in bioinformatics and computational biology at the University of Minnesota. She is also an attending neurosurgeon for Centracare and the Minneapolis Veterans Administration Medical Center. Today, she'll be helping us to understand the future ways in which we'll diagnose and treat brain injuries. Thank you so much for joining us today, Dr. Samadani. I'll turn things over to you.
Well, thank you so much for inviting me, Amber. I am thrilled and delighted to be part of this. I think it's incredibly important to talk about brain injury because it is what we call the invisible injury, the injury that nobody can easily measure and therefore treat. So I'm absolutely delighted to be invited, and I'd like to thank you guys for giving me this opportunity to participate. Without much further ado, I'm going to go to my first slide. Let me hit share screen. And is that working for you? Yes. Okay. So basically, as you mentioned, I'm a neurosurgeon. I work at the Minneapolis VA Medical Center. I'm an associate professor in the Department of Bioinformatics and Computational Biology, and then I spend the rest of my time working as a neurosurgeon at Centracare. So I'm pretty busy. These are my, let me go to the next slide here. These are my disclosures. So I have intellectual property related to concussion and brain injury assessment that I will be talking about. Also to assessment of the chronic consequences of brain injury and to treatment of intracranial hemorrhage. I won't be talking about the bleeding part. I'll just be talking about the assessment parts. The other side of this slide is my grant funding, my salary, employment, consulting fees, honorariums, or equities. I just mentioned this so that nobody can say that I didn't claim, or disclose all my conflicts. But in terms of this talk, most of this stuff is not actually relevant. The one thing that is relevant is that I will be talking about an eye tracking technology. This is FDA cleared. But the way I'm going to discuss it in this talk is not currently FDA cleared nor commercially available. So I'd like to start by talking a little bit about conventional approaches to science versus machine learning. So 100 years ago, the way we did science was we made a hypothesis, and then we tested that hypothesis, and we saw how well it worked, right? Well, things have changed. Now we use something called machine learning. Machine learning is observation-based. What this means is we don't necessarily make a hypothesis or even try to fully understand a model. A machine comes up with an explanation of data that may or may not have fully understood components. It doesn't require human understanding. And then we test that as an independent data set. What this means is that we can sort of use the machine to learn, and so that it ultimately mimics human intelligence, and that's called artificial intelligence. Where is this useful? Well, many people don't realize this, but every time you go to the grocery store, many chains, many different types of stores have face recognition software. And what they do is the minute you walk in the door, they identify you. They know how many visits you've made to that grocery store in the past and how much money you spent last time you were there. This type of software is used in airports, it's used in public spaces, it's commercially available, And it reduces the number of patrols that have to work in a particular area. They use it for concert security, for example. So can you imagine if you used this kind of technology for things like brain injury? You know, you can imagine that it would dramatically change the way we treat and recognize brain injury. Imagine if every time you walked into the emergency room, instead of, you know, getting conventional things done, you could have this much knowledge about someone right away. So I would propose that radiology will be one of the first fields that will be impacted by machine learning and artificial intelligence. Already there are automated ways of reading these scans such that the machine has an earlier ability to detect changes than would be previously anticipated. So there's algorithmic analyses that are done. And that can tell you not only about things like stroke, but they can also tell you about things like swelling in the brain. Here at the University of Minnesota, we have the tremendous privilege to have Dr. Camille Ugerbil. He's one of the scientists working on the human connectome. And what that is a sort of a project looking at pathways and connections in the brain. And they've published their algorithms online. Anybody can have access to them. And then you can apply them to different problems. So you can look at connections in dementia, or you can look at connections in Parkinson's. And in our lab, we look at connections in brain injury. And this is work that was done by one of my graduate students. And she's looking at 3 different types of analyses of an MRI scan that can tell you about things like integrity of the brain, or cellular breakdown, or axonal damage. And each of these things has an algorithm that allows you to calculate how intact these pathways are. And what we find, and this is an abstract that she presented at a meeting, was that when you compare trauma patients to control patients, there are fairly dramatic differences in many of these metrics. And so what this is this is not a radiologist saying, hey, you know, this scan is different from that scan. This is an algorithm saying, hey, this scan is different from that scan. And so what you can imagine is that at some point we'll have algorithms that can differentiate a trauma patient from a normal patient pretty much completely on its own. Or, you know, a Parkinson or an Alzheimer patient, you know, different from a normal patient, you know, a normal person, completely on its own, independently, algorithmic analysis. So I would say that's the first step. Physical examination is something else that can be made sort of algorithmic and objective. So eye movements. Eye movements are something that you don't necessarily think about. Eye movements are controlled in the brainstem. So you think about, you can purposefully look in a particular direction, but the fact that your eyes move in a coordinated fashion is not something that's conscious. It's something that's controlled in your brainstem. And what that means is that when someone has a brain injury and the pathways that are feeding into the brainstem are disrupted, you can detect it by measuring the coordination of eye movements. And that's something that my lab has worked on quite a bit, and we've started a company in this space. So here's an example of someone, he's actually a researcher in my lab, who's looking at a computer screen, and the computer screen has an aperture in it, and the aperture is showing a cartoon. In this particular case, it's a Disney movie. And what he's doing is he's watching that Disney movie as the Disney movie moves inside the aperture around the screen, and we measure his pupil position. And what that tells us is how well his eyes are capable of moving and how coordinated his eye movements are. And that can reveal important details about function of the brain. So we can tell when there's swelling in particular areas of the brain, or we can tell if certain nerves are disrupted. So if you disrupt the nerve that moves the pupil up and down, then the pupil doesn't move up and down. If you disrupt the nerve that moves the pupil out to the side, then the pupil doesn't move as well out to the side. So when someone has a brain injury, they hit their head, for example, the pressure in their head may go up, and the function of the nerves in the head may be decreased. And we can measure that by looking at changes in range of movement as the person watches a computer screen. And so what we see is that in a normal person, the right and the left eye move together. And in a concussed person, they may not necessarily move together. So this can be a powerful diagnostic for brain injury. And what we've shown is that as people recover from brain injury, they tend to get better, and their eye movements tend to trend towards the same types of eye movements as normal people who are uninjured. And this can be useful for not just emergency department settings, but also sports medicine settings. What about blast injury? So blast is when something explodes and the shock waves travel not just directly impacting the skull, but directly through the skull. And there are multiple mechanisms by which that can happen. And so many of you will remember Minnehaha Academy. So Minnehaha Academy had an explosion, right? And there was a large blast, and there were a number of survivors. They were evaluated in our lab with eye tracking and with other measures. And what we found was that their eye movements were different. There were 5 metrics that were significantly different between the people who survived that blast and other people who were nowhere near that site. And when you look at the map of the school, the blast happened at the yellow site. The 2 people who died were in blue. And the more abnormal your eye tracking, the brighter red the circle. So the people who were closest to the blast had the most abnormal eye movements, whereas people who were further away from the blast had the most normal eye movements. So one can use this type of technology to potentially ascertain whether or not someone has had a brain injury in ways that are not typically measured. So this is just a summary slide of the different aspects of brain injury that you can assess with something like eye tracking. Well, what else can you do? So you've got radiology, which tells you what the brain looks like. You've got eye tracking, which tells you how well the brain functions. And now you have serum markers. And serum markers, they're proteins that are found in the blood after a brain injury. They can tell you where in the cells in the brain the damage occurred. So there are 2 major classes of cells in the brain. There's glia and there's neurons. And they have different proteins that are expressed on their surface. And when someone has a brain injury, those proteins may cross the blood-brain barrier and be detected in the blood of that person. So you can send that blood to the lab and you can check for protein levels. And what we see is that at different time periods after the injury, different proteins are expressed in the blood. And so we can use all of this information to classify the nature of an injury, right? So a spontaneous bleed in the brain may result in certain expression of certain proteins. Denying the brain oxygen can result in expression of other proteins. Stretching or tearing neurons can change the expression again. And then having a CT scan that shows a vascular injury or is negative for a vascular injury can also change expression of proteins. And then you have people who are uninjured and you can compare them. And ultimately what you can do is you can combine all these things. So you can combine things like the CT images or the algorithmic analysis of CT images. You can combine the eye tracking. You can combine the serum markers. And then you can look at the genetics of that person. And then you can sort of put that all together and figure out how to treat the brain injury. So this table is a summary of some of the genes that we know are involved in brain injury. And some of these are pre-injury. So what that means is people who express these genes are more likely to sustain a brain injury. They're more likely to engage in risk-taking behaviors. There are certain genes that predict whether or not someone's going to be a mountain climber, for example. And those are called pre-injury genes. Then there are genes that immediately after a brain injury are implicated in poorer recovery. And then there are genes that are associated with the chronic effects of neurotrauma. So a higher risk for something like dementia after a brain injury. So the goal is to take all of this information, to take all of these objective measures and put it together to create an algorithm that will enable us to classify the nature of injury and then treat it. And so that's really what our lab is focused on. So ultimately, I would say the robots are coming. It's going to begin with diagnostics, physical examination, radiology, and pathology. And then it's going to, so you have something as, complex as a concussion, and this is a schema of the multiple pathways that are involved in brain injury when someone has a concussion. And you can see that it's super complicated, and it suggests that there are multiple places that things can be disrupted. Why is this helpful? Because this is a complex problem. One of the biggest mistakes people make is they assume that a problem or a particular solution will be simple. People had assumed that with dementia. They thought that dementia was due to deposition of amyloid protein in the brain. But what happened was drug after drug after drug that was targeted at treating amyloid deposition failed. And why is that? It's because the problem is more complicated than just a single pathology. And so the ultimate take home here is when you can't accurately measure and classify a problem, don't count on being able to treat it. We don't want to make the same mistake with brain injury that had previously been made with dementia. Let's see, next slide. And this is one of the papers about the number of failures. This was published in the New England Journal of Medicine, and you can see that every single one of these circles is a medication that has failed to treat dementia. And the problem is that we don't want to replicate this error with brain injury. We cannot assume the problem is simple. I believe that big data will help. The more data that we have, the better. These are the risk factors for dementia. This is from a review published by The Lancet. And you can see that there are a number of risk factors that are responsible for a huge number of cases. So having less education early in life, hearing loss, high blood pressure, obesity, smoking, depression, physical inactivity, social isolation, and diabetes. Together, collectively, this accounts for approximately 35% of cases. So a third of all dementia could be reduced or prevented by controlling for these risk factors. That's another, the point here about dementia is that, you can use big data to predict who has a higher risk. This is a study of 41,000 patients, 284,000 controls, and they figured out that certain medications increase your risk. So this is the kind of data that we need to understand brain injury. Ultimately, I think that if we have objective measures and machine learning, we will be able to figure out the acute and chronic effects of neurotrauma. And so then we'll be able to control them. So neurotrauma basically means, someone gets hit and their spinal cord or their brain suffers consequences. And, these consequences are really diverse. You can have spinal cord injury. You can have, damage to the endocrine controlling parts of the brain. You can have damage to the inner side of the ear that controls balance. You can have electrolyte disruption, which causes headaches, seizures, stroke. And then you can have the full spectrum of neurosurgical problems. You can have stretching or tearing of neurons. You can have a lack of oxygen to the brain. And ultimately, if you can diagnose and classify the nature of injury, the goal is that you would be able to treat it. So my hope is that someday brain injury will be classifiable. When we combine objective assessors and machine learning, and then we can use that information to prevent the chronic consequences. Those are the people in my lab. And thank you. I think I can take questions.
All right. Thank you so much, Dr. Samadani. I am waiting for just a few questions to come in. I'd like to just remind our audience that if they do have any questions, that we do have some time to answer them, that they can go ahead and type those in the box, and we'll spend some time answering those questions. I had a couple that I was just, that I thought about just in listening to you describe the work.
Yeah.
You had mentioned early on that a lot of these algorithms are essentially open source. They've been published and they're available for a variety of uses. Obviously, this combines a lot of disciplines. It combines, disciplines in the medical field. It combines disciplines in computer science and in mathematics, which, it's always interesting to talk about the intersection of those things in the modern way that we treat individuals. Can you tell us a little bit more about your interest in that intersection or the groups and teams that you work with to make this possible?
Yeah, I think this is a really, really valuable point because probably, 50 or 100 years ago, you could be a specialist in a field and make advances in that field. And in my opinion, that's not the case anymore. Now, the way to solve big problems is with collaboration. You need people who have a skill set that's completely unique from your own. Like, I have some limited understanding of brain injury, but I have extremely limited understanding of computer science or machine learning, relative to my colleagues in bioinformatics and computational biology. So I collaborate with people. I collaborate with Yuk Sham and Roy Kwang at the university. They are both professors who help me to develop the algorithms that will enable us to classify the nature of injury. Because I think that, my skill set is completely different from theirs. And without combining them, there's very little probability of getting great solutions. All of my work in my career that I can think of that's really been impactful has been through collaboration with people who are often from extremely disparate fields. So yeah, excellent point.
I appreciate that. I think that's universal across a lot of fields. That collaboration is really necessary to solve, like you said, these really big problems. One of the other questions that I had is in talking a lot about this need or this hope to be able to classify the nature of the injury, you talked a little bit about limiting failures and providing better sources of treatment. Can you also speak to a little bit about how you might actually think that would impact the quality of care that patients are receiving?
Yeah, so right now, someone gets hit in the head, they go into the emergency department or, wherever they go, primary care physician, and there are very limited tools that are universally accepted as diagnostics. I mean, the main one is the CT scan, which is an imaging test. So that we can tell a little bit about the CT scan, but if you have a normal CT scan, we know very little about the rest of your brain injury. There are some research MRI sequences that might be helpful, but we do not have a current standard of care that is fully diagnostic. And so this is in complete contrast to any other organ system in the body. Like if you break a bone, you go to the emergency department, they do an x-ray, they say, your bone is broken, we're going to fix it, right? Very, very, very straightforward. When someone has a brain injury and you don't see anything on CT scan, it is not straightforward. Getting it diagnosed, getting it treated is not straightforward. And so we need to make brain injury just like every other organ system in the body. We need to be able to measure function. We need to be able to assess, chemical disruption, physical disruption, physiologic disruption. And we need to put all that information together and create a picture of what that brain injury is like so that we can treat it. Because until you can diagnose accurately and classify the nature of the problem accurately, you'll never be able to treat it. And that's the mistake that's happened with dementia, is that, characterizing the nature of cognitive decline is so complex that we don't even have great scales for it. And therefore, we've failed in developing therapeutics for the same problem. And, this is something that is not solvable with single molecule pathways. You can't assume that it's simple. And so I think, conventional science has served us extremely well for the last 1,000 years. But now, in order to solve these bigger problems, the brain problems like, headache, chronic fatigue, dementia, and brain injury, just among many, many, many others, there are other problems as well, but just those four, to solve those four, you're going to need a lot of data and a lot of information and a lot of understanding of complex pathways.
You know, as we look forward to the future and trying to solve these big problems, you know,
Yeah.
hopefully we have a lot of students who are either in a program right now or who are younger and are, you know, trying to explore their career pathways. Can you tell us a little bit? We at the Bell Museum are always interested in people's origin stories and what got them interested in the research that they're doing. Can you tell us a little bit more about what led you to the path that you're on now and the schooling that you acquired to make that happen?
Yeah, absolutely. So I knew I wanted to be a doctor from an extremely young age. I think, you know, basically from kindergarten, because I always wanted to help people. And I was always very curious. You know, like if I saw an ambulance, I was always wondering, you know, what's going on in there, and I wonder if that person's going to be okay. So I wanted to be on the providing end of healthcare, you know, my entire life. I picked neurosurgery because I saw that you had an opportunity to have very immediate gratification. Like if you help somebody with a neurosurgical operation, they get better very quickly. And it's really very, very rewarding. I ended up in brain injury basically um, because I was working at the VA hospital, and our patient population is so frequently exposed to brain injury. It's got the highest per capita incidence of any, you know, subset of the American population, that you really realize what a profound problem it is and how few answers there are. You know, when I meet people with brain injury, almost universally, every single one of them will say to me, I sometimes feel like I'm crazy because, you know, nobody can measure what's wrong with me. And therefore, you know, I feel like, is it real, or am I, you know, is there a psychiatric component? You know, is this a real problem, or am I just crazy? And I feel like we need to get rid of that. We need to make brain injury just like a broken bone. We need to make it something that you can measure, you can see, you can treat. And I've sort of been obsessed with that idea for the last, I would say, at least 15 years. And I'm hoping that we're moving in that direction. You know, for your viewership who may be potentially younger and interested in neuroscience fields, there's so much opportunity here. You can literally pick anything, you know, related to mental health or wellness, and there's a lot that's unknown and a lot of opportunity to make a big difference. So I think that it's a very open field, and there's a lot going on that could potentially, you know, be dramatically improved upon.
Thank you so much for sharing that perspective and for sharing that encouragement for younger students. I want to thank you very much, Dr. Samadani, for joining us today. I know that you were in surgery earlier today, so thank you for taking time out of your schedule to help provide some of this education. Yep. I want to thank also our audience for joining us for today's installment of Brain to Brain. Once again, special thanks to Boston Scientific for their support of brain-related programs and activities like this throughout the Bell Museum. We hope that you can join us tomorrow at 11 a.m. for the next program in the series. We're going to be actually exploring some brain imaging technology and how it's being used to improve diagnosis in autism. So a little bit different than what we discussed today. I will post a link to the entire Brain to Brain schedule of live programs in the comments box below so that you can explore that and see what we have coming up. Thank you once again, Dr. Samadani. I hope everyone has a wonderful evening.